Okay, is this your experiment?
No, that's Bruce's. That's this.
This reminds me of your grandfather's lab.
This is my experiment.
Oh, here's your desk.
Yeah, that's my
Yay.
desk.
Apoptosis. This is cell death, right?
Yes, apoptosis.
Apoptosis.
Yeah. This is mine, or the results of mine, and you can see. So, just to explain, this is the one that worked but didn't really work, and this is one that never worked at all.
Oh.
That's the B is the negative, or is the positive, and the A is the negative. If you just look now, obviously it's like a day after, but if you look at the difference, the color difference, this is negative, this is positive, this is negative, this is positive. They're practically the same in this test and vastly different in that test. This one, the kit works. Now it didn't give us good results because the other results are so low, like really low. But this one just doesn't work at all. Everything pretty much equalizes out and becomes the same thing after like 30 minutes. Which is annoying. That's Dr. Maxwell's office.
Oh, okay. Cool.
Yeah, these are this is water.
Oh.
We have to autoclave the water, and we get it from a special filter.
Oh, okay.
So
it's special water.
Yes, it's special water. That is probably the purest water you will ever drink.
Well,
Either
you don't
that
drink
or...
it.
Well, you can.
Well,
Well, not
yes.
that, because autoclaved. This also is water.
Uh-huh.
Right here, it's,
Hey, this jar has wheat in it.
yeah, it does. It's weird.
That's
kind
funny.
of odd. Yeah,
Is
the
there a
safe-
company that...
This is the safe lab, and yes, we do use
The
it.
safe lab? This is the
We do
safe?
make milk.
No.
No. Like, it's as in
not
like,
dangerous.
not deadly. Although none of the labs are deadly
Right.
because you can't catch
Instant
cancer.
milk?
That is used for this.
Oh.
This is what's called a blocking buffer, and it's used in Western blots for the secondary antibody and washing and complicated stuff. But
I
it's
see.
not just milk, it's like chemicals and a soap-like substance.
Oh, okay.
So I would not drink that because...
No. Obviously not. It's got
And
lymphoma
actually-
in it too, right?
No, that's not that.
Oh, okay.
Fortunately.
Here's some, is those photos or negatives or something?
Those are photos, yeah. So
Okay.
that's why they call them blots.
Oh, I see. That's the Western blot.
Yeah, and the grad students having difficulty to see this right here.
Yeah.
Those should all be about the same level. Same with this, and this, and this,
Uh-huh.
and they're not. So
Oh, okay.
he has to add different concentrations of samples. The next lab, I don't know if Bruce is in the lab, so... Oh, that's our fridge also, by the way.
Okay, but not for lunch.
No.
Yes. Well, this...
So this is the lab that don't touch anything.
No, I'm not touching a thing.
I know, but this is particularly the lab. The other lab you technically can't eat, but there's nothing in there. So I do actually eat lunch in that other lab. There's nothing in there that's deadly. Not even the chemicals are really dangerous. They just...
Uh-huh. But this lab is different?
This lab is different because...
This is where the human cells are?
Specifically this room,
Ah.
which there's the primary view, but that's, yes, it's ultraviolet light and it kills cells.
This is
Kills
where you
them.
kill your cells?
This is where we do anything and everything with the cells is done
Okay.
in that fume hood.
I see.
So usually, oh, I'll turn the light on. Don't touch me.
I'm not touching it.
Usually,
So it's...
just the glass system. But usually this is how.
You work with your hands under...
Yeah. And you have to work in, and you don't want to move your hands like this because it disrupts airflow.
Which you just did.
That's okay though. And every time anything happens in here, you always spray. This is 70% ethanol.
Oh, okay.
And then,
Is it closed all the way?
yeah, when it stops buzzing, it's closed. There's a little
There's a
bit
little
of
gap?
a little bit of a gap, but that's fine because
Oh,
the air
okay.
doesn't go through there. It goes down into that grill. It gets sucked in. by that grill.
I see.
And then This is the microscope. Easy. Fridges, freezers.
Oh, it's stereo.
Yeah.
Cool.
And here, I'll show you.
Is this your stuff?
This is all of the cells, all of the cell lines, the different cell lines that we use. Most of this stuff is Bruce's stuff. This is apparently a cell line set up by the medical student, which is also here for the summer. These are Dr. Maxwell cells, and the cells that I mainly use for my experiments, G3 and 2631. They're in big flats because we need lots of them. The nice thing about these cells is they're free-floating. So these cells are stuck to the bottom of the plate. They attach.
Yeah, okay.
It's not agar. It's a liquid. It's a liquid medium.
Those are free floating.
These are free floating in the medium. So you don't need to do. You don't. With this, you have to add a chemical called trypsin, which detaches them from the bottom of the plate. I don't know, you can kind of see there's like little spots and stuff on the bottom of the plate.
Okay.
But these are just free floating, so
these are human cancer cells.
Yeah, these are human cancer cells.
Okay.
Now, luckily, we cannot catch human cancer cells. You cannot catch cancer from other cancer cells.
Right, your immune system
Yeah.
would recognize it as alien.
Yeah. Now, there's one more thing I want to show you. This. So we have a very specialized way of counting cells, incredibly high tech.
1,
2, 3,
Really?
4, 5, 6.
You actually count them. You eyeball them?
Yep. See those little grooves?
Yeah.
You basically pick one of those squares
Yeah.
and count how many cells are in that square,
Oh, in
and
the
you do that.
in the microscope.
Yeah, and you do that like 4 times.
And then you average.
And then you average that, and then sometimes even 8 or more.
I see.
Yeah, but yeah, this is the don't touch anything lab.
I didn't touch anything.
I know you did. And that's bleach. We use bleach to kill the cells. We need to. That's a centrifuge. That's a bath. But yeah, that's pretty much it.
Cool.
Yeah.
Really cool.
So most of about
pretty
half
exciting,
of my
huh?
work is very exciting. Half of the work that I do goes on in this room, half in the other room. This room is, if I ever work with cells, this is where I do it. Nothing. Most of the rest of the stuff is all Baru's doing stuff, the grad student. The other room is for anything. It's what you use when you're not working with live cells. Anything else goes on there pretty much because it's a lower level.
This is the grad student's desk?
This is the grad student's desk. Yeah.
Okay.
He's Iranian, so.
Okay. All right.
Yeah.
Is that it?
Pretty much. I guess I can show you my light state. That's literally frozen things.
What's that?
I said, I guess I could show you my lysates, because I've pretty much frozen things and tested in here. Lots of things. Lots of things.
I see.
These are all mine.
Not for food storage.
Those are mine. That's mine. That's mine.
Okay. Cool.
Yeah. And that's where we keep liquid nitrogen.
Apoptosis or necrosis? Necrosis is different?
Yes. Apparently.
Okay. Apoptosis is when the cell intentionally kills itself?
It realizes that there's a harmful thing inside of itself, and if it partitions itself off into smaller groups, then it can be eaten by macrophages and stuff like that.
Oh, so
Necrosis,
it intentionally destroys itself.
Necrosis is different.
Necrosis is when it just dies
It is, yeah.
on its own. I mean, without intending to die.
Yeah. That's the cryogenic tank.
Interesting.
Every week you have to fill it. And
So this is your professor's office here?
yeah.
Oh, okay. Is this his lab office, or does he have another? He has another.
I think
office?
this is his like office. I don't know.
Okay.
I think he might have another office in the new building, but he mostly spends his time here.
Okay.
Yeah. It's not very big, but it doesn't have to be.
Yeah. Cool.
Yeah. All right.
So it's you and one grad student?
Yes, and then one med student for the summer that's not part of the summer research program but knows Dr. Maxwell from my classes and stuff like that.
Okay.
Wanted to just sort of help with his research.
Hey, you get your own desk.
Yeah, I got here first, so the med student is over there, and I'm right here, and I feel pretty happy.
Hey, you know, I have a nameplate that says T. Davis.
Really?
Yeah, I can give it to you. You could put it up here on the... You want to?
For the 3 weeks I'm
For
here,
the
yeah,
3
sure.
weeks? All right, I'll
last
dig it up.
Yeah.
I'll dig it up. Where are we?
There's the autoclave room. Those are distilleries for our distilled water we can pump lots of places. The special part about this room is the ice machine.
Oh, not for human consumption?
No.
Does that mean that this doesn't eat people?
Yeah.
I will consume humans.
But, yeah, those are the autoclaves. This one is fun to operate because to open it you have to crank. I can't open it now because it's in the cycle, but you have to crank it. I have to slide it, and you pull it up.
So where's the any key? It says, I don't see a key that says any. Oh, it's computer control, huh?
Yeah, even the trash has to be autoclaved. The
Wow.
biohazard waste.
Yeah.
Yeah, so it's good one. The glucose 6-phosphate dehydrogenase pathway and the reversal of drug resistance in lymphoma cells. The enzyme glucose 6-phosphate dehydrogenase, G6-PDH, converts glucose 6-phosphate into 6-phosphogluconate, initiating the pentose phosphate pathway. It has been reported that higher activity of the pentose phosphate pathway is linked to a higher expression of the NRF2 gene and a resistance to CHOP chemotherapy in several different types of cancer cells. We investigated the role of G6-PDH pathways in mediating resistance to CHOP chemotherapeutic regimens in lymphoma cells. 2 cell lines were studied, a CHOP-sensitive 2631 cell line of diffuse large B-cell lymphoma and a derived CHOP-resistant G3 cell line. It is hypothesized that G3 cells have higher activities of G6-PDH than 2631 cells. The results presented here support this hypothesis. When G6-PDH is inhibited, the G3 cells become more sensitive to the CHOP drug regimen.
Timothy Davis.